All right, so I have the pleasure of getting to introduce our OutSmarting 2023 recipients. This is one of the highlights of our conference where we get to present the awards in person, and we have our family friends here to present some of these awards. So I wanted to just give a quick overview before we get into the fun stuff on what OutSmarting is. So OutSmarting for us is a really meaningful grant process. And so there are kind of a few things that we think about as we put together what our process looks like. One is that we really want to successfully integrate both scientific rigor as well as patient-family voice. So we have a peer review process, but we also have stakeholder reviewers in our process. So we have some family funds eligible to be stakeholder reviewers depending on how much money they've contributed to the grant, and we don't expect them to, score on methodology or rationale or anything like that, but they are uniquely positioned to be experts on patient impact, and so we think it's very important to include that in our review process. Our grants, as you'll see, are also a very meaningful way for our families to honor their osteo warriors and angels. We have several awards this year that are fully funded by some of our family funds, who you'll meet in a second, and it's also really important as to foster young talent. We're so pleased to have so many young investigators with us this year. We started a Young Investigator Award last year. It's a $50,000 annual grant, and so you'll see that we have a couple of Young Investigator recipients this year. And another thing that's really important to us in this process is that we facilitate future funding. So if you apply to OutSmarting, we have these amazing peer reviewers who take the time to provide excellent constructive feedback. And we think it's really important to share that back out with our applicants. So if you apply to OutSmarting, we'll provide that constructive feedback. We don't, we want great osteo projects to get funded. Even if we don't have the budget, we want it to get funded elsewhere. And so we're happy to help facilitate that process. And the other thing that's really important to us is accountability for our donors, for our family funds, for their donors. So it's really important for us to have reporting. So we have reporting at the mid-month and also at the end of a project milestone. This is just quickly our funding priorities. So we're really focused on late-stage translational research, studies with promising preclinical data that can move forward to clinical trial, or also specific funding gaps in clinical research, and you will see an example of that this year. And quickly, our focus areas, which is pretty much everything. The only thing that we don't really focus on is survivorship and supportive care. So this year, 2023, we have $600,000 in grants, which is amazing. Last year, we had $450K, which we thought was unbelievable. Yeah. So as I mentioned yesterday, since inception, we funded over $1.5 million in osteosarcoma research. This year, we are so excited to fund 7 scientists, 2 of which are young investigators, supported by 47 family funds. So thank you for our family funds also. All right, so we're gonna get started with the fun part. I just had a kid graduate today, and I'm like, this feels like the class of 2023, OutSmarting, please rise. No, I'm just kidding. So we are gonna start first with Amanda Marinoff from the University of California, San Francisco. Thank you, Sherrick. And so Dr. Marinoff is going to be studying toward precision oncology, developing novel molecular biomarkers for risk stratification in pediatric osteosarcoma. And Dr. Marinoff's award is because of Charlotte. So I'm going to have Charlotte's dad, Sherrick, present Dr. Marinoff's award.
Hi, my name is Sherrick Marinoff. I serve on the MIB board. I'm here at my 3rd Factor conference, so excited to be here again. But today I'm representing my family on behalf or because of Charlotte. Charlotte was diagnosed in 2018 at the age of 15. Many of you know Dr. Sweet Cordero. He was Charlotte's primary oncologist at UCSF Benioff. Despite many trials and treatments and such, Charlotte became an osteo angel in September 2021. She was very brave, she was very fierce, she was a fighter. She had a great smile, but she loved MIB agents. She served on the MIB junior board for 2 years. She actually created her own clothing line and sold it online and donated the profits to Charlotte's Family Fund. So today we're very grateful for our friends and family that we're able to provide a Young Investigator Award to Dr. Amanda Marinoff.
Thank you so much, Sherrick. This is remarkable. This award is because of Charlotte, and as I shared with Sherrick yesterday, and I'd like to share with all of you, I will never forget Charlotte's radiant smile, her unparalleled sense of spunk, and the quizzes that she would make and hang on her hospital door room and make all of the doctors and nurses take before she would let us examine her. This award is also because of all of my current patients and my future patients, and it's because of all of us. What am I actually going to do? Well, we heard yesterday from an amazing panelist about a lot about the heterogeneity of osteosarcoma and the critical need to develop biomarkers for risk stratification, and that we're developing a sophisticated understanding of the biology underlying subsets of this heterogeneous disease. And the goal of this project is clinical translation. How can we get these biological insights to patients? So in my first aim, I'll define a transcriptional signature based on RNA-seq of a small subset of highly predictive genes that will allow us to distinguish between prognostically distinct subgroups of osteosarcoma using about 100 primary patient samples with rich clinical annotations and paired RNA-seq data. And the hypothesis that's really behind this aim is based on really exciting work generated in the Sweet Cordero lab, and you'll hear later from Dr. Lopez Fuentes, who's really the hands and brains behind this work. In the second aim, I will develop and validate, based on this signature, a clinically feasible targeted gene expression assay in order to bring risk stratification into the clinical trial setting and ultimately to clinical care. The platform that I'll be using is a nanostring platform, which we heard a little bit about yesterday, with the idea of developing a tool that is easy and fast to run and to analyze, is relatively cost-effective, accessible, and has flexible sample input. And the overarching goal of this work is to be able to ultimately, in a clinical trial setting, and if validated in clinical care, be able to use this test up front to ask a patient diagnosed with osteosarcoma, are they likely to respond to MAP, or do we need to use different therapy? And initially that would likely be on an experimental arm of a high-risk trial, and then ultimately move toward risk-adapted biology-tailored therapy, really moving away from the one-size-fits-all treatment model and moving us to a precision-based approach. So I want to thank Dr. Alejandro C. Cordero, who's my primary mentor, as well as Dr. Katie Janeway, who's my former mentor and current collaborator in this project, who's going to be sharing samples with us. How lucky am I to have two of the most amazing mentors on both coasts. I'd like to thank the entire Sweet Cordero lab, especially the powerhouse group of women scientists who are here with me today and who will wow you throughout the day. And to MIB agents for all of the extraordinary ways that you really are working toward making it better for osteosarcoma. Thank you to you all, and thank you again, Sherrick, Ali, and Matt, for this extraordinarily meaningful award. Okay, next, we have another Young Investigator Award for Marta Roman Moreno from the University of California, San Francisco. And Dr. Roman Moreno is going to be studying CRISPR screening to identify vulnerabilities in metastatic osteosarcoma. And Dr. Roman Moreno's award is because of Sydney. And I'll invite the Kaplans, Greg and Sarah, Sydney's parents, to say a few words.
Thank you, Christina. Thank you, and good morning, everybody. So my name is Greg Kaplan. This is my wife, Sarah. Our daughter, Sydney, was diagnosed in 2018, fought for over 2 years, and passed in November 2020. This award, our family fund, is simply because of her, but she was also very outward-looking and really cared about others. She absolutely loved this conference, and when she was here at HQ, she was worried about the other kids and took care of the younger kids, so I love that. There's another girl in our town who is struggling right now, and this is also for her. So Special thanks goes to one particular donor who made this possible. So a special shout out to the Patel family in New York, very dear friends of ours who funded the entire thing. So again, special thanks to them. And that's about it. So, so we'd love to present this check.
Thank you.
It's already endorsed on the back.
Should I move it?
I think-
Okay.
You can take that from you, you don't have to hold it.
Thank you so much. Hi, thank you everyone. My name is Marta Roman. I'm a postdoctoral researcher at the Sweet Cordero Lab at UCSF. It's a pleasure to be part of this wonderful and inspirational initiative. I'm so grateful for this award that I hope will bring the cure of osteosarcoma a bit closer to the patient. It's my first factor. I hope it is not the last, because so far it's been an amazing experience. So I hope to be able to come back next year and show you my results that I hope will be promising for osteosarcoma. So my work is focused on metastatic osteosarcoma. The title of my project is CRISPR-AI screening to identify vulnerabilities in metastatic osteosarcoma. To do this project, I'm taking advantage of three main very valuable resources that we have in our lab. First, we have a very decent amount of patient data available, both gene expression data and DNA accessibility. Second, we have generated a huge panel of patient-derived xenograft model and cell lines. Finally, we also have an in vivo orthotopic model of metastatic osteosarcoma that I will use to study the metastatic process, since this model mimics really well what happened in patients regarding metastasis. So my first aim is to characterize the role of selected target genes in the onset and maintenance of osteosarcoma primary tumors. And the second aim is to identify drivers of metastasis using a CRISPR-AI-based genetic screen approach in osteosarcoma in vivo models with PDX-derived and mouse osteosarcoma cell lines. And how I'm going to do this? So first, I have already generated a list of genes taking all these patient data that I mentioned before. So this list contains genes that are specifically related to metastasis in osteosarcoma. And the main objective of the project is to test the role of these metastatic-related genes in the onset of metastasis using different PDX cell lines that are representing the heterogeneity of osteosarcoma in our in vivo model. So we hope we will be able to identify new potential vulnerabilities for the metastatic disease, bring one of these targets for a clinical trial, and see if we can help this wonderful osteo warrior here. So thank you very much. I'm so grateful for this. I'm so happy to be here with all of you.
We were just discussing over there who was going to be next, and it's Dr. Ryan Roberts. Those are our young investigators, a hand for our young investigators. And we have $500,000 awards this year. And so the first is Ryan Roberts from Abigail Wexner Research Institute at Nationwide Children's Hospital. And Dr. Roberts is going to be studying combination therapies targeting heterogeneity and lung environment in metastatic osteosarcoma. And this year we have a funding partner, Vision for the Cure, represented by Tracy Russo, who's here with us today. We're so happy for her to be here at her first factor because of her brother, Matt. So Tracy, do you want to come up and?
As she said, good morning. I'm Tracy Russo, and I'm here on behalf of Fishing for the Cure. My brother Matt was diagnosed at 18 in 2003, and 5 years later, he, being a young adult, he was like this, adamant that this can't continue to happen for the younger children. So it was his dream to help find a cure, and that was a promise I made to him before he passed in 2010. And we started Fishing for the Cure in 2008, and we continue today. So thank you to all of our sponsors and donors who have continued to support us throughout the year, the many years we've been around. We're super excited today and honored to be here to award Dr. Roberts with this.
Well, thanks to Tracy and her family for all the work that they've done to help us to work together towards the shared goal of making it better for all of our patients with osteosarcoma. I think in my lab, we've chosen to focus on understanding better what it is that makes osteosarcoma grow within the lungs, but really not other places as much. And in the study of this, we've kind of learned a couple of key lessons. And one of those lessons is that when tumor cells go to the lung, there's really a stereotyped process, a developmental process by which these tumor cells are able to establish a place where the tumor cells can grow and become clinically relevant metastases. And as we've studied that further, we've understood that actually there's different types of tumor cells within a primary tumor that execute different programs during that metastatic development. And as we've kind of delved into those processes, we've found that there are some early steps that are really important that are mediated by cytokines and by interactions of specific types of tumor cells with the surrounding lung epithelial cells. And one of the consequences of these interactions is to set off a wound healing cascade that ultimately creates an environment where the tumor cells that grow very rapidly can establish a home and can ultimately kill our patients. And so we've been trying to find ways that we can block different elements of this process and have kind of stumbled on some combinations that we think might work. And it's kind of coincidental in a way that some of those combinations are combinations of drugs that we already use in kids, largely for other diseases, and combinations that coincidentally might work together in ways that might counteract each other's side effects. And so our initial experiments in some mice where we just tried it once to see were really promising, and this grant will let us explore that both mechanistically and functionally a little bit further in our animal models to see if this might be a viable treatment strategy for preventing and maybe even treating lung metastasis. And so we thank MIB agents and Fishing for the Cure and for the way in which Tracy and her family have chosen to honor Matthew and his goal, which we share to make it better for kids in the future. So thank you.
We're very casual here. Okay, our next award is for Heather Gardner from Tufts University. And Dr. Gardner is going to be studying advancing liquid biopsy platforms to support early detection of metastasis and inform future interventions in osteosarcoma. And Dr. Gardner's grant is actually a team effort. Like many things we do here at MIB, it is because of Annalee and also because of Fishing for the Cure. So Tracy, I'll come back up here, and also Annalee's parents, Debbie and Pierre, to present Dr. Gardner's award.
Hi, my name is Debbie Grillo, and this is my husband, Pierre, holding the big check. Our daughter, Annalee, was diagnosed, excuse me, in August of 2018. And her osteosarcoma was very rare. It was beta HCG positive, and there is no data and no information about beta HCG positive except for like 3 anecdotal articles I found online. And so we're really excited about MIB and the work that they're doing. And Heather's work, Dr. Gardner's work that she's doing in discovering the metastasis before they get so far along that it's just too late to do something. And we're really excited about Dr. Sam, I'm going to mutilate your last name, Vochenbaum? Sounds good. I'm sorry, excuse me. Work that he's doing collecting data, because by the time that we were looking for a clinical trial, we were in full-blown COVID, and it was really searching in the dark, and we felt very alone. And so the work that all of you are doing is so important. And when our daughter died, we went on her phone, and she had created a will. And it was very simple. I want all my clothes to go to children in need, and I want all my money, and God bless her little heart, she saved about $5,000 from babysitting and other odd jobs that she did. I want all my money to go towards osteosarcoma research, because I don't want any other child to have to suffer the way that I have suffered. And that is why we are here, and why all the work that all of you are doing is so powerful and important, and it really does make a difference. So thank you all. Thank you, MIB. Thank you, Anne. What you have created is amazing, something out of nothing that is really making a difference. So thank you, everybody, and thank you all for the work that you're doing.
So I am incredibly grateful to have the opportunity to honor Annalie's memory and all the other osteo warriors and osteo angels. So again, I'm a veterinary oncologist, and as you've heard today, we see a lot of similarities in dogs as compared to those that we see in kids. And one of the things that I've been really interested in is metastasis, and understanding the process of going from the primary tumor to the metastatic lesion. And one of the challenges that I saw both in our dog patients and that I was seeing in kids was that we were kind of waiting for things to happen. We would have biomarkers that we could look at early on, but we couldn't really track things longitudinally. We kind of had to wait to see disease progression. And so when we started looking into liquid biopsy, we said, oh, that's really interesting. So we can look for the disease early on in the blood, but is there more that we can do? And so the goal of this work is to look at multiple parameters in the blood and use a computational platform, so a machine learning platform, to try and identify signatures of metastasis. And in doing this, we'll be using banked samples from dogs that we have obtained throughout their disease course, from the time that they're diagnosed with the primary tumor to when they develop metastatic disease, to really identify signatures that differentiate the primary tumor from the metastatic disease. And then in the second part of this effort, we're going to do a prospective study in dogs where we treat them with neoadjuvant chemoimmunotherapy. And we track them with liquid biopsy throughout their disease course and try and identify signatures that early on, within a few weeks of starting therapy, can tell us, are they responding the way we want them to? With the goal of ultimately being able to give opportunities to change therapy early on before we've developed gross metastatic disease. And so, again, I'm really honored to have the opportunity to pursue this work. And really am looking forward to being able to share with everyone, hopefully next year, an update on how this has progressed. And again, just incredibly honored for the opportunity. Thank you.
Thank you so much to Debbie for just really giving us an example of when we say meaningful grant process, that's what we mean. It's like, as you can see, it's a very powerful award that you guys are getting. So with that, I'd like to also introduce Sam Volchenboom from the University of Chicago, who's going to be using his grant to establish a data commons to support new treatments for osteosarcoma. And Dr. Volchenboom's award is because of Annalie.
I'll be quick. We took pictures like half hour ago outside, and that's when I learned. I said, "Oh, parents can say something." I'm like, "Oh, great, okay." So I'm like, "Well, I know my wife's gonna say something. She's, she can do something really quick, really fast." But when our daughter was sick, I watched my wife and a good friend of ours that is a doctor searching everywhere for treatments. It looks like when we call hospitals, nobody knew what was going on somewhere else. We called, outside the United States, it was some research were the same, which seems strange for a medical field, something that is high-tech. It looks like there was missing gaps, and what we finding this year seems just simple. It's like collecting data, so then someone can go look at it and say, "Oh, this may work, this may not work with what I have here." But I really wanted to thank my wife because she has done all the work. She really did. Thank you.
Hi, so that's a hard act to follow. Listen, we started Data for the Common Good with the goal of making the world smaller, bringing the world's data together. And data are all over the world in silos, in databases, in registries, email attachments, paper binders, you name it. And with a disease like osteosarcoma, it's too rare. You can't have the data sit around like that. You have to bring it together. And what our group has done is we work on better ways to share data. And we devote ourselves to building a data dictionary, which allows us to take all the data and put it into a common model. And then we make all this data available back to researchers so they can use it. When I was a resident back in the 90s, for a child with newly diagnosed ALL, there was only 4 kinds of ALL. You were boy or girl, and your white count was either greater or less than 50,000. And now when a new kid presents with ALL, there's a dozen different pathways they go down based on all these diagnostic criteria and different ways we stratify the kids. And that's because we collected data on tens of thousands of kids, and we were able to understand better which kids need more treatment and which kids need less treatment. We need to do that for osteosarcoma. And we're going to do it by bringing lots of data together. We're going to expand our data dictionary to include biomarkers and genomic elements and molecular data. And we're going to get people to share. We don't take no for an answer. When we first started this, looking at osteosarcoma a couple of years ago, people said, "Oh, no one's going to share with you. Everyone's too competitive." And here we are a couple years later, and we have a fully grown consortium with members in the US and Europe and all over the world, and people are starting to put data in, and we're starting to collect it, and we're going to make those data available to researchers. We also use that same information to power a clinical trials matching platform, which allows clinicians and families to come in and find clinical trials for patients based on those clinical and molecular factors. And it all gets back to this idea of community building and sharing. So I'm just so thankful that you recognize the power of what we can do together. I'm so honored to get this grant, and it means so much to me and our team to be able to do this work on behalf of Annalise. So I'm just so grateful, and it's just wonderful. So thank you to MIB agents, and to Ann and Christina and everybody else. We're just very excited. So I'm looking forward to talking to you all afterwards, and thanks so much.
Okay, so we're still giving out money. So our next two grant recipients are actually funded by all of our family funds. And so you heard Anita talking yesterday early in our MIB panel about fundraisers and events, and you heard Mary Basaga talking about their family fund and how they raise money. And so these are all our family funds who are selling gold bows in September. They're doing outrunning events. They're having a bake sale, or, it's grassroots fundraising that all these families are doing to have supported these next two grants. So first we have Dr. Shahab Asgarzadeh from Children's Hospital Los Angeles, and he's going to be studying the identification of circulating immune signature as a biomarker of disease response and resistance in patients with relapsed osteosarcoma treated with nivolumab and regorafenib. And this is an example actually of a correlative study that we're funding. It's actually a correlative study for SARC-038, and we're so pleased to be able to support that. So Dr. Trucco is going to present the award on behalf of all of our family funds to Dr. Asgarzadeh. If you want to come on up.
That was a pre-COVID picture, so I really appreciate the families for giving us the opportunity and the funding to do this trial. This is my first time in the factor meeting. I've enjoyed it, and I've really been inspired to continue to do some of this work. I come from a different group of researchers, but they told me last night that once I start doing osteosarcoma, I remain doing osteosarcoma. So I will continue. So there is plans for doing it. So I'm going to explain a lot of the words that you see on and get a little bit more lay language. First, the award is on behalf of myself, Dr. Fariba Naveed, and the team that we have assembled, including Laura Davis and Troy and Brian in Harvard. So in the sum, what this grant is all about is trying to figure out why certain patients respond to the therapies that we give them. When we start a clinical trial, we're really waging a battle against the tumor. We're arming the body with drugs and methods to try to kill the tumors and also empower the immune cells, which are the soldiers in the body, to go and kill the tumor cells. If you just wait and see what happens to the patient at the end, you see the battles are sometimes won, sometimes lost. There are some changes. You won't really learn what's happening. It's like giving soldiers the tools that they need, and then not going to the battlefield and looking and see what happens and how they actually do their job. So the correlative studies that we do, we collect blood a lot of times, we collect tumor tissues. It's really for us to get a snapshot of what's happening in the battleground when we arm these soldiers and we attack the tumors. So these two drugs that, I don't know who they come up with these names, but regorafenib and nivolumab are basically two drugs that we actually give to the patients. And one of them basically goes in and tries to cut the supply lines for the tumors. And the other one, it also actually builds roads for the soldiers to get into the tumor site and do their job. And the other one, nivolumab, actually helps some of the specific type of soldiers, T cells, to remain not exhausted, to remain active when they get to the battlefield. So, and in this trial, we've actually seen some patients that have a really durable response to these therapies, and some patients who, as if like you're giving them water, and we don't know why. And this is really crucially important for any clinical trial that we do in osteosarcoma. We have to collect tissues, we have to collect blood, we have to collect plasma at regular times to understand why this is happening. And why is this important is because it allows us to figure out what went wrong in some patients and what we could do better for them. So the two trials, the two assays that I do to look at these things, we collect blood, and we look at what's called the RNA sequencing. I look at all the marching orders for these immune cells that are given out, and read them and basically see if there's any marching orders in patients that respond to the particular therapy that could give me hints of what I could do to improve the therapy. And also look at using a CyTOF, I look at the types of immune cells, which regimens have gone up in numbers, which type of T cells are there, what type of B cells are there, what are their numbers, is there a difference between patients who actually responded to a therapy given these drugs versus not. And that gives us ideas to go back to the clinic and bench and use different types of methodologies to improve our therapies. And once the trial is, once all the data is analyzed, I'm going to give all the data to Sam. He's going to put it in his database, and we're going to learn more from adding more data to it to figure out what the next trial should be. So with that, again, I'm grateful for the support and the collaboration, and thank you all for listening to this brief talk.
And then our next OutSmarting recipient is Robert Cantor from UC Davis. And Dr. Cantor is going to be studying inhaled canine IL-15 to maximize immunotherapy responses in dogs with metastatic osteosarcoma. And Dr. Cantor's award is also because of all our family funds. Congratulations.
I forgot to take my beta blocker this morning, so you'll have to bear with me. And I promise this is not an Oscars speech, but I tend to ramble, so I wanted to make sure I had my notes in front of me. It's truly a great honor to be here for all the reasons you've been hearing about over the last few days, and I'm very grateful. These types of grants can be very transformative, and they can really make a difference in sort of the research trajectory and pursuing projects that you're passionate about, but may have difficulty getting funding from other mechanisms and so forth. So I'm really excited for this project, and I'm really excited and grateful for this opportunity. Our proposal is based on our prior work. You've heard a lot about the comparative oncology over the last 2 days, and that's been very exciting, and we've been a part of that. And one of our initial canine immunotherapy studies was actually repurposing a sort of drug family in terms of cytokine therapy, and delivering it in a somewhat novel way in terms of an inhalational approach to deliver it directly to the site of action in terms of lung pulmonary metastases, but to limit systemic exposure and hopefully try and improve upon the risk-benefit profile of cytokine therapy. And we were excited because we did see some significant responses in that trial, but we were giving recombinant human IL-15 because we had access to it from the NCI biorepository, and it's a CTEP available agent. So we can only give it for 14 days, and because of risk of cross-species differences and the risk of neutralizing antibody formation. So we were excited about that, and that led us to our hypothesis that if we were giving recombinant canine IL-15, we could give it longer, we could maximize responses. With immunotherapy, responses can sometimes be delayed, and potentially improve upon the benefits we observed. And so that's the background for this project, and we're going to be using recombinant canine IL-15. We're going to be giving it basically until progression or unacceptable toxicity, and we're going to be assessing that in a phase 2 Simon two-stage approach to see if we can really advance this as a therapy and potentially translate it to humans as well. And so the clinical endpoints are in AIM-1, And in AIM-2, we're trying to build on our immune monitoring correlative studies. We use flow cytometry, RNA sequencing, and in our previous trial, we've observed that there is immune effects in the circulating immune populations, especially the sort of business end, the natural killer cells and the cytotoxic T cells, indicating that we are seeing some immune effects of the therapy, and we'd like to further explore that and really try and drill down on predictors of response and potentially biomarkers. And in addition, we've had some new preliminary data showing that some of these receptors, such as the IL-15 receptor alpha, can be expressed on suppressive cells, and so again, trying to really improve upon kind of stratification and biomarkers to really understand the mechanisms of response and resistance to therapy. And so that's the substance of our proposal, and I'm really excited, and I really, again, couldn't be more grateful for this opportunity. Thank you.
Okay, Dr. Marinoff actually had to dash off to a family member's wedding, so we're actually so thankful she made the effort to come out here. But I present to you the class of 2023 OutSmarting. We're actually a little ahead of schedule. So do you guys have any questions for any of our OutSmarting recipients? Oh, question over here?
I just have a rather specific question for Dr. Cantor. Is the therapy, is it like an inhaled, like the vaccine for dogs, or is it like a albuterol kind of,
like a nebulizer.
Yeah. Okay, yeah.
Yeah, we have a cute video on our website about a dog getting the treatment, and it's very well tolerated, actually.
How long does it take?
It takes about 15 minutes, okay, each one. We mix it up in a saline bag with albumin and just deliver it through one of those little nebulizers. And they have to have a cone over them, obviously, because it doesn't just stay there. But yeah, it's tolerated well. And then we send them home with the drug. At least, it's stable for 14 days, so they can keep it in the fridge or, in a cooler. And, we'll have to work out some of the more logistics for longer duration of therapy, but that's how we did it.
Very cool. Thank you.
Hey, this is a question for Sam, and I guess pretty much for everyone up here. When we're thinking about these, the preclinical trials that we all do in mice and some of the correlative trials, the parallel patient trials that are done in canines, what's your thought about building a database where we can actually harmonize that data as well? So rather than trying to go through PubMed or, let's be honest, Google now to see what mouse work has been done and so on and so forth, actually having another data set that is kind of parallel to what you've built, so that we can harmonize all of this data so that we have a centralized place to look for preclinical studies, human studies, canine studies, kind of like a one-stop shop that kind of takes the burden off of us as researchers spending hours scouring through things and actually just trying to have a very singular solution for that.
Yeah, it's a great question, and I'd be interested in hearing others' thoughts too, but we've talked a bit about having a preclinical data commons, what it would look like, how we would get the data, how it would be consented, how we would get the data sharing in place, and I think it's something that we could definitely do. We'd have to understand what other repositories are out there for PDX models and things like. There's a lot of different databases out there. Actually, there's a canine data commons that already exists. It's part of the Cancer Research Data Commons ecosystem, and as I understand, they have a very limited amount of canine data in there. And so we'd be happy to help understand how to get more of the canine data in there so that we can actually federate those sources and make it available to researchers, even through our interface. And for any data that doesn't have a home, we're happy to take it and to harmonize it to a standard and make it available. So if folks want to talk about a specific kind of preclinical data, we'd be happy to take a look at that and help set something up. But the most important thing is to get a lay of the land first to see who has preclinical data sets and how we would put those data in. That's a great question. Anybody else have thoughts?
I mean, I would just say it's a good idea to start with first things first, and I think that one of the really transformational things that Sam's group has done is just to give us the language so that we're all using the same language, which then makes it easier to harmonize things, even animal studies in the future, if we're all using time points that match and the same words for certain types of failures and progression. That facilitates that going down the road.
And any excuse to work with dogs is okay with me.
Rob is getting his steps in.
Thank you. Just an update, just to follow up on the ICDC, the Integrated Canine Data Commons, so that is up and running. You're correct. There is a relatively limited amount of data in there. We are looking to fill that up. So if you do have canine data that you would like to deposit in ICDC, please contact me and I can put you in touch with Gina Cuffle and Jim DeTillo, who are involved in uploading that data, harmonizing it, data curation and uploading it. So just a shout out. If you have data, we are looking to increase the data amount in there. Oh, I'm sorry, Nicola Mason from Penn.
That's great. So you just need to know who to ask, and she's right here. Thank you.
Okay, great. Thank you so much. I would love to introduce Dr. Chan Khanna, who is the Strategic Advisory Board for our friends and colleagues at the Osteosarcoma Institute, who's going to talk a little bit more about all their research funding initiatives. But I just hope that you have a couple takeaways from this panel and presentation here, which is we actually had a discussion group yesterday called Turning Pain into Power, and it was patients with osteosarcoma who enabled research. And in that context, we were talking about sharing data, sharing tissue. And I think clearly you can see it's in another context here where they're actually really fundraising and working hard to financially support research. So thank you again to Family Funds for doing that. And I think the other takeaway, and you'll hear more about this from Dr. Khanna, is that this disease community is a very enthusiastic and passionate patient community that's really excited to partner with the scientists to help fund research and get that done. So I hope you leave with that today. So thank you, Dr. Khanna, for...
Christina, thank you again for a fantastic meeting. And I think the C in factor is collaboration. And as a result of the Osteosarcoma Institute's valued relationship with MIB and making it better being a big thing, we are very focused on concentrating on the cure. And that creates a great opportunity for us to all play on our strengths and work together. And in the spirit of collaboration, we're very excited to be part of this with MIB. So I am representing the Osteosarcoma Institute. Chelsea reminded me to say that we do have a table up front if you'd like to learn more about the Osteosarcoma Institute. Within the Osteosarcoma Institute, the focus, or my responsibility is really to help shape the research agenda within our Strategic Advisory Board. See, the Osteosarcoma Institute was started in 2017 for many of the reasons that we've heard already, and really not having treatments for patients was unacceptable to us. And so we assembled a board of advisors, and this board is not just scientific, it's strategic. And I'd say that at the high level, we are trying to balance a responsive to a prescriptive approach to doing research, and that's part of our agenda as we concentrate on this cure. Collaboration is definitely the foundation of achieving this. We believe that we are the source of questions, not answers, and we seek partners to find those answers. Through the work that we've done since inception, we've committed $5.2 million to osteosarcoma research, so we're excited about that.
Great. And then our next OutSmarting recipient is Robert Cantor from UC Davis. And Dr. Cantor is going to be studying inhaled canine IL-15 to maximize immunotherapy responses in dogs with metastatic osteosarcoma. And Dr. Cantor's award is also because of all our family funds. Congratulations.
I forgot to take my beta blocker this morning, so you'll have to bear with me. And I promise this is not an Oscars speech, but I tend to ramble, so I wanted to make sure I had my notes in front of me. It's truly a great honor to be here for all the reasons you've been hearing about over the last few days, and I'm very grateful. These types of grants can be very transformative, and they can really make a difference in sort of the research trajectory and pursuing projects that you're passionate about, but may have difficulty getting funding from other mechanisms and so forth. So I'm really excited for this project, and I'm really excited and grateful for this opportunity. Our proposal is based on our prior work. You've heard a lot about the comparative oncology over the last 2 days, and that's been very exciting, and we've been a part of that. And one of our initial canine immunotherapy studies was actually repurposing a sort of drug family in terms of cytokine therapy, and delivering it in a somewhat novel way in terms of an inhalational approach to deliver it directly to the site of action in terms of lung pulmonary metastases, but to limit systemic exposure and hopefully try and improve upon the risk-benefit profile of cytokine therapy. And we were excited because we did see some significant responses in that trial, but we were giving recombinant human IL-15 because we had access to it from the NCI biorepository, and it's a CTEP available agent. So we can only give it for 14 days, and because of risk of cross-species differences and the risk of neutralizing antibody formation. So we were excited about that, and that led us to our hypothesis that if we were giving recombinant canine IL-15, we could give it longer, we could maximize responses. With immunotherapy, responses can sometimes be delayed, and potentially improve upon the benefits we observed. And so that's the background for this project, and we're going to be using recombinant canine IL-15. We're going to be giving it basically until progression or unacceptable toxicity, and we're going to be assessing that in a phase 2 Simon two-stage approach to see if we can really advance this as a therapy and potentially translate it to humans as well. And so the clinical endpoints are in AIM-1, and in AIM-2, we're trying to build on our immune monitoring correlative studies. We use flow cytometry, RNA sequencing, and in our previous trial, we've observed that there is immune effects in the circulating immune populations, especially the sort of business end, the natural killer cells and the cytotoxic T cells, indicating that we are seeing some immune effects of the therapy, and we'd like to further explore that and really try and drill down on predictors of response and potentially biomarkers. And in addition, we've had some new preliminary data showing that some of these receptors, such as the IL-15 receptor alpha, can be expressed on suppressive cells, and so again, trying to really improve upon kind of stratification and biomarkers to really understand the mechanisms of response and resistance to therapy. And so that's the substance of our proposal, and I'm really excited, and I really, again, couldn't be more grateful for this opportunity. Thank you.
The mission is simple, and that's to dramatically increase the options for patients and deliver survivorship to patients. And that's through the funding of the work that we do. And as I said, that funding agenda comes from both a responsive approach, which is receiving grants, but then trying to be a little bit prescriptive in terms of identifying and fostering the right questions to deliver these improvements in survivorship. And this slide shows our board both in person and virtually as we meet as a team quite regularly. The Osteosarcoma Institute works within these committees, and the committees are shown in different colors. In green, we have the Strategic Initiatives Committee, which really tries to do many of the things that were discussed, but says, how do we help bring new technology to clinical trials to answer biological questions in patients in clinical trials? So correlative science being a big part of that. And sorry, I misspoke. That's the blue to the right. That's Brian Crompton and Richard Gorlick, who lead that scientific initiative, correlatives committee. In the light green in the upper left is the work that Bill Tappan and do, and that the idea there is simply to say, rather than a formal grant process, can we deputize ourselves to find good ideas and really through discussions provide resources, meaning funding, relationships, or domain knowledge to bring people, especially from other areas like malaria research, and bring their research to osteosarcoma when it's appropriate. Obviously, an important focus in a committee that does the work in development, and that's related to the team at the Quad W Foundation, so we're close partnership there. And then a clearinghouse role, hopefully trying to bring biopharma closer to patients and clinical trials in osteosarcoma, and that clearinghouse function is now being taken over by Dr. Laura Davis. It's great to interact with her at this meeting here over the last few days. Just to quickly bring attention to the specific studies. So the translational studies, they're listed here. I'll just give a quick tagline to each one. Dr. Gorlick's work is around antibody drug conjugate development, so developing new conjugates and bringing them into the clinic through translational work. Around the concept that osteosarcoma is a disease of altered transcription, Pete Scacciuri, in the work of metastasis, is developing a therapeutic strategy on this inhibition with CDK inhibitors. Ryan Roberts' work that he described to sort of dig into how cells get to the lung, how they cooperate with different populations of cells to produce metastasis. And then a comparative oncology agenda with the University of Minnesota, focusing on natural killer cell, NK CAR cells, combined with a radiation therapy for osteosarcoma patients. So that's both a dog study in development and a human clinical trial. These translational studies include work of Dr. Eustine, who's here today, and that involves his sophisticated mouse models and the use of combinations of immune therapy to deliver improvements in outcome. And Dr. Sweet-Cordero looking at how the STING pathway, which is supposed to detect these abnormal karyotypes in osteosarcoma and kill those cells, but for some reason doesn't do that in osteosarcoma. So how do we activate that pathway so that pathway does what it should do? Dr. Weiss, good friend of mine and long-term colleague, who has a very sophisticated model of bone development in osteosarcoma and can track the transition from normal bone development to osteosarcoma and metastasis within this sophisticated mouse model. Really exciting new work that Kurt has advanced. The correlative studies, again, leverage the idea that we need to do research within human clinical trials and bring new technologies to these trials. Brian Compton doing that within circulating tumor cells in patients in clinical trials. And you heard a little bit about the work yesterday at the University of Florida with nanoparticle development. And our work here is to build on the correlatives to optimize the vaccine strategy in osteosarcoma. We do have a very strong interest in supporting clinical trials that are obviously in the clinic, and Dr. Livingston's work is very focused on combination approaches with immunotherapy, realizing that immunotherapy as single agents has not given what we want in patients with osteosarcoma, so this is iteration around combinations. Dr. Janeway's work is around the idea of how do we target this abnormal, complex gene structure, which is probably connected to some DNA damage response. So how do we perturb or mess up that DNA damage response in osteosarcoma patients with new drugs? We have in the balance between receiving grants and being responsive to good ideas and telling people what to do, we've tried to find a happy medium and convene meetings on specific topics of interest to us. And the recent one was a genome symposium, which asked, how do these cancer cells shatter chromosomes so quickly? And then how do those cells survive? They shouldn't survive. And how does that lead to opportunities for translation? That's a completed meeting, and we have some actions now to follow up from that meeting. And in the fall, we'll be having a meeting to study what's on the surface of osteosarcoma cells, since it's been such a struggle to really understand recurrent events within the tumor cells themselves. And then with an understanding of what's on the surface, we can develop different approaches towards therapy, drug conjugates or CAR T cells or what have you. So really, thanks again to MIB for all of that you do in making it better. We're trying to do it in a similar way with a focus on this concentration on the cure, and these are the families and the patients that we seek to also provide improvements for. So I'll take questions if I can.
Any questions?
So I'll offer an addition. So this one committee that's called SIFT, there's no grant involved, there's no submission, it's really just a phone call. And if we can, through a phone call, say, this is a gap that we need to address to bring your work forward, to compete for a grant within MIB or elsewhere or within the OSI, we'll try to do that together. And that's just a phone call. So please reach out to me, to Chelsea, come to our booth, and we'll figure out how we can connect to provide that gap. It's not just money, it's relationships and resources that we have to accomplish that gap-filling exercise.
Thanks a lot, Dr. Hanna, for your presentation. Do you fund only US-based researchers, or how about Canadians? Do you fund anybody in Canada?
So I'm a Canadian, so yeah. But to answer, yeah, we do fund outside of the US. We've funded projects in Europe, in Canada, so yes.
Can I ask a question real quick?
Please.
So as you're looking at these studies and thinking about how the data are going to be collected, if anybody wants to work with us to take the standards we developed to help inform the way that the data are collected, then the data can be what we call born interoperable, and to be much easier to deposit into the commons to use later. And we'd be happy to advise any group that's going to do a human study on data collection forms and how best to collect the data.
Great, well that's very helpful, I appreciate that. And as you know, Dr. Janeway is part of our team, and so we are aware of the data commons connection, so I'd love to do that. And I want to also just bring attention to so the data is collected in the way that you've described. There's also a way to visualize all of this data. It's called the Osteosarcoma Explorer. So the Osteo Explorer is a web portal where data has been aggregated and visualization tools have been developed, and hopefully we can find a way to have those two things come together, because the visualization tools are really made for sort of less sophisticated people like me to ask, does this protein do something? Is it important? Can I introduce you to a drug company? It's less about the sophisticated knowledge that you and Troy put together to dig into the data. So the visualization tools are for the less sophisticated groups in the audience who want to join me to try to understand this and really ultimately bring people who are developing drugs to osteosarcoma in a rapid manner. So I encourage you to look at the UT Southwestern portal, Osteosarcoma Explorer. Osteo Explorer is really what it's called.
We have another question. Down in the
Dr.
front.
Kahana.
Straight ahead, Doctor.
Here. Just a quick question. So what's being discussed within the symposium, would that be available to the osteo community?
So the complex genome meeting, so ask Chelsea, is that, so I'm sure we'd love to make it available. I don't answer the answer of how, but maybe Chelsea, you know if it's recorded. The findings will certainly be available on our website. I don't know that there is video content. I don't know that that video content can be shared, but certainly the findings, and the findings are next steps, which mean what do we do, how do we support efforts to dig into this complex genome. Those will be the next steps, memorialized in the form of RFPs and specific investments in research.
Any other questions? Thank you very much.
Thank you. Thank you.